disclosure, the things I'm going to talk about weren't done by me. They're done by some really impressive people in this room, including Drs. Hirosh, Diamond, Goel, and many others, physician scientists. So it's really a pleasure to be here and to try to give some context to the clinical problems that you are experiencing. And so in this disease, we have an amazing array of problems that affect all of the parts of the body. And as you can see from this diagram, there are essentially every single system in the body is affected. And I'm sure when Drs. Erdheim and Chester were first seeing these patients, they were kind of mystified. How can one disease, or apparently by the histology at that time, how could one disease affect all of these organs? And they were aware of cancers, which would metastasize, say, from the kidney to the lungs or the liver, or maybe leukemia or lymphoma, which would be concentrated in the bone marrow, lymph nodes, spleen, and perhaps liver, and occasionally the CNS, but really never go to the heart or the kidneys. So I think this provided a great conundrum to them. How could you explain a disease that would go so many places? And over the years, the work of many investigators in this room and elsewhere have shed some light on the type of cells that cause this disease. And that's my mission today, to kind of explain, where did the cells come from that cause ECD? Well, they come from the bone marrow. And this is a picture of a bone marrow biopsy from a normal person. And all these little purple and dark dots are the cells of the bone marrow. And the very darkest black dots are the red cells and the early red cell precursors, the nucleated reds. And then there's some sort of purplish cells that are kind of dark purple and have nuclei that are big and open, and those could be the histiocytes. But you know, this doesn't really tell us where the ECD disease comes from, and so we have to go to kind of a little bit of a summary diagram here, which shows the sort of the growth parameters of the hematopoietic system. And so you start at the top with what's called a hematopoietic stem cell. And between 1 in 10,000 and 1 in 100,000 cells in that bone marrow aspirate or biopsy that I showed you would be a stem cell. A stem cell is the great grand person cell. It makes everything. It can go to the platelets, it can go to the white cells, it can go to the red cells. And how it gets there is really a fascinating story. And so these stem cells not only have to go many different directions to make the cells that help us live, but they have to renew themselves. So that great grand person can't go away. They have to maintain themselves throughout our lives so that we have a continual supply of blood cells. And that is a really incredible thing. So we're going to end up down at the bottom of this diagram in the dendritic cells and the macrophage area, because those are the cells that cause Erdheim-Chester and the other histiocytic diseases. But how we get there is really kind of a fascinating story. And so we, like everything in medicine, we go from baby steps to baby steps, and we eventually learn some new things. And what we learned back in around the 2005 era was that there was some common myeloid progenitors and some common lymphoid progenitors, which kind of took that stem cell down one road, and that road divided into different pathways. And so at the time, about 20 years ago, it was thought that Langerhans cell histiocytosis came from a distinctly different lineage of cells than the Erdheim-Chester. And you see the kind of the blue and orange dots as they go down the pathway there. And the interesting thing was, it was understood that if you went a little bit to the right-hand side to the yellow monocyte cells, that if they received the stimulus of a couple growth factors, transforming growth factor beta and interleukin 4, that these cells could then switch over to another pathway and kind of find themselves into what would be the Langerhans cell histiocytosis or the Erdheim-Chester. And so all of this was possible because of some incredible technology called flow cytometry. And that is the process whereby single cells are put through a little stream of fluid, and those cells have some little tags on them in which laser light can excite those tags and emit a red signal or a green signal, an orange signal. And those signals will tell the investigators, is this a cell of the monocyte lineage or the histiocyte lineage, or which part of the histiocyte lineage? And as the eras have progressed, we've gone from just a few dozen to now hundreds of markers. And so there are flow cytometry systems now which can literally look at hundreds and hundreds of markers on the cells and figure out where is this cell in its progress through the lineage of this journey towards maturation, and where did this cell come from? And so there are experiments in culture, not in the person, but in the culture, where they can put these growth factors and develop these cells into the different families and say, oh yeah, this one came from such and such a beginning place. So it's really been quite an interesting journey. So before I get a little deeper into this cell of origin talk, I think we have to branch off a little bit into the mutations that cause these histiocytic diseases. And many of you have heard about the BRAF mutation. Well, if you look at the left-hand side of this photograph, and there's a normal situation where a growth factor docks or joins up with a receptor on the surface of the cell. And this is a normal function where a growth factor is supposed to help a cell grow from a very early white blood cell to a later stage white cell, for instance. And that growth factor docking with the receptor sends a signal down through this pathway. And you can see it goes from RAF to RAS to RAF to MEC to ERK. And then that circle at the bottom is supposed to imply the cytoplasm of or nucleus of the cell, and the signal has come from the cytoplasm into the nucleus of the cell, and that little arrow that goes down to the horizontal arrow and the squiggly DNA thing is trying to tell us that a piece of information is going to the DNA of those cells, telling it to grow or to differentiate or do something that's good. So this signaling process only works for a short time. And so the on switch at the top turns the on switch of each of the next steps. And finally, it gets down to the nucleus, and that on switch says, do this, and then the switch is off. Well, if that pathway is subverted by a DNA mutation, which takes that on switch to an always on thing, then the signaling keeps going. And so that little second horizontal box of the RAF box is there because there's an ARAF, a BRAF, and a CRAF. And the particular mutation that is associated with many Erdheim-Chester patients is the BRAF V600E. And if that mutation happens, then that signal is always on, and that signal is telling the cell, keep doing this, do this, do this, do this, ad infinitum. So what does it do? Well, you see on the far right-hand bottom there a couple of things. One arrow's up, one arrow's down. And the arrow that's up is a gene called BCL2, which prevents cells from dying. And this is really an important part of this whole process, that these cells that are part of the immune system, they're supposed to do their thing, then get out of the way and die. Well, they don't. They stay there. And the reason they stay there is because of the other arrow, which shows that there's something called CCR7, and that's down. So CCR7 is a molecule, it's called a chemokine, and it directs cells to move. Only here, they're not moving because the chemokine isn't there. So the cells are staying in one place, and that's why they cause rashes, holes in bones, collect around kidneys, et cetera, et cetera. And so all the bad things that happen are because this BRAF mutation subverts the normal growth and function of these cells. So Paul Milne and a group from Newcastle and some collaborators from around the world took this to another level, and they looked at the cells in the blood of patients with Erdheim-Chester disease and Langerhans cell histiocytosis, and they found that there were some common things that happened. And so the LCH cells kind of went from a common dendritic cell precursor on the left and made those blue cells in the blood that are circulating, and they are stimulated by a growth factor called GM-CSF and TGF-beta, and they become the LCH cells. Now, there's another pathway in the middle, the CD14 monocytes, which they originate from the stem cells the same way, but they've gone down a little bit different road, and they become the monocytes that have a surface marker called CD14. And if you treat them with a growth factor called Notch, then it subverts them over into the LCH pathway. But if you treat them with another growth factor, MCSF, then they become the ECD cells. So what's happened here is that cells that started as a stem cell became somewhat intermediate type of maturation of a cell, and then they differentiated into cells that cause these different diseases. So you can think of this in a more sort of global pattern, that these cells, these white blood cells, get into the bloodstream, and because they have the BRAF mutation, they do bad things. And the BRAF mutation probably is not in the stem cell, because if it were, then every cell would be differentiated into these malignant cells. It's probably that mutation happens at a little bit later stage, so that some people get diseases just in their skin and their bones, other get kidney, other get heart, and it's this differentiation of the cells by the different cytokines and the different maturation markers that allows the cell to cause the problem. So they get into the bloodstream, they infiltrate tissues, and they become these foamy cells that are surrounding the kidney, and they're giving off growth factors which cause all the problem. So just a little diversion here into chemokines, and this is an overwhelming slide. I mean, I can't keep track of all these things myself, but kind of the bottom line is that chemokines help cells move to certain places. And so there are hundreds of different combinations of these markers on the surface of the cell, and some of them would direct the cells to go to the heart, some to the brain, some to the skin, some to the kidney. You get the idea. So they can go lots of different places, and that's why Erdheim and Chester were so mystified that why could a cell go to all these places? Because leukemia cells don't go to all those places, but Erdheim-Chester is a special case that it goes to these unique locations, and the cells stay there and they create problems because they are giving off some growth factors, which are inflammatory growth factors, which create all the problems. So, you know, the signs of Erdheim-Chester disease include fever, fatigue, muscle wasting, foggy brain, all kinds of kind of how do I feel symptoms. And those how do I feel symptoms come from these cytokines, which are produced at excessive levels. They're normal molecules, but there are just too many of them. And those signs and symptoms are the reason, are because of these cytokines swimming around. So it'd be nice if the cytokines did just their own job and make a cell attack an infectious particle, but in this case, they're kind of attacking the body. They're accumulating in places where they're not supposed to be and causing heart failure, kidney failure, lung failure, brain fog, and brain trouble. So it's quite a mess of chemokines and cytokines that the stew all together, these molecules create the disease that we know as Erdheim-Chester. So I think I'm just about up to time. I got 1 minute left, but I think I'll stop and I'll open the floor up for questions. Yes.
And if y'all have a question, make sure you raise your hand so we can bring you the mic, or that
Dr. McClain, I think you answered the question, but why not stem cell transplants?
So could a stem cell transplant be done for Erdheim-Chester patient? Under certain circumstances, maybe if there were organ failure that was only partially stopped by the therapy. But the problem with transplants for patients who have diseases like leukemia and lymphoma and Erdheim-Chester is you have to make sure that you've gotten rid of all the Erdheim-Chester cells. And that's kind of a big question, because if you treat with inhibitors, they are marvelous in decreasing clinical symptoms, but if you stop the inhibitors, then the disease comes back. So I think we're at a point where the stem cell transplant question has to be first, the problem has to be solved with getting rid of all the bad cells.